Now the next is Clostridial fermentation. Clostridia are anaerobic bacteria that ferment a wide variety of carbon sources including glucose, galactose, cellobiose, mannose, xylose and arabinose. Clostridia are capable of utilizing a variety of free sugars and carbohydrates. Though several carbon sources such as glucose, xylose, various starches, corn molasses, cheese whey and even algal biomass have been employed in the past as AB fermentation substrates, and the secretion of various enzymes by this bacteria are documented. The focus is now shifted to renewable and sustainable non-food raw materials such as agricultural waste. These feedstock is primarily lignocellulose, which implies that the current ABE strains cannot directly ferment them due to want of cellulitic machinery. Here lignocellulose refers to plant dry matter, that is biomass, so-called lignocellulosic biomass. They are basically used for the production of biofuels, mainly bioethanol. So it is imperative to use pretreatment and hydrolysis steps for the generation of fermentable sugars from the biomass. However, this process also generates a lot of compounds that can be inhibitory to the clostridial growth and ABE production. The current butanol producing Clostridium species naturally produce acetone along with butanol and ethanol, that is ABE. Acetone production by solventogenic Clostridium species is regulated at both genetic and protein levels through the expression of acetoacetyl coenzyme A (CoA) or CoA transferase coat, as well as acetoacetate decarboxylase ADC, with the combined action of these enzymes resulting in the production of acetone. Here solventogenic Clostridium species have a biphasic metabolism composed of an acidogenic phase and solventogenic phase. During acidogenesis, these bacteria are able to convert several carbon sources into organic acids, commonly butyrate and acetate. The AB fermentation is a biphasic process that converts sugars into acids, that is acetate butyrate, and solvents acetone, butanol, ethanol. Biofuels such as ethanol and butanol and several chemicals such as acetone 1,3-propanediol and butyric acid can be produced by these Clostridium organisms through fermentation processes, among the well-known species known as Clostridium carboxydevorans C. This is the image of Clostridium carboxydevorans C. Now, this is a simplified metabolic pathway of Clostridial ABE fermentation. Here, biomass like cellulose, hemicellulose, lignin undergoes upstream processing. Upstream processing comprises tasks in the initial stages of the fermentation process. This includes all steps related to the development of microorganisms, nutrient preparation, cell culture, cell separation, and harvesting. Upstream processing involves nutrient preparation. This upstream processing forms sugar, example glucose, xylose, mannose, arabinose. Now this undergoes fermentation leading to the formation of pyruvate with the release of CO2 and H2. Now this pyruvate is converted into acetyl CoA. Along with acetic acid, it forms ethanol. Again this acetyl CoA is converted into acetoacetyl CoA and form acetone with the release of CO2. Now this acetoacetyl CoA converts into butyryl CoA along with butyric acid to form butanol. That's all. Thank you.